Good afternoon. My name is Aditi Ravikumar. I am an undergraduate student at the Dementia Lab. For the past 2 years, I have been studying a case of early onset Alzheimer's disease. The proband began showing the onset of forgetfulness at the age of 38. He was a Caucasian male with 10 years of education. He was finally diagnosed with AD at the age of 43 when he went into the clinic concerned about developing AD because he had a very significant family history of dementia, as we can see in the maternal pedigree on the slide. At the time of diagnosis, his MMSE was 25. As the disease progressed, his memory deteriorated, but he also began to experience symptoms such as dyscalculia, aggressiveness, irritability, and anger outbursts. Eventually, he developed flat affect, apathy, anhedonia, and by the age of death, at 47 years old, he required significant help with activities of daily living. He was unable to drive, and he had significant problems speaking and comprehending. After death, genetic analysis revealed a G to A transition in exon 7 of the presenilin-1 gene, predicting an amino acid substitution of glycine to glutamic acid at residue 209 of the presenilin-1 protein. No other mutations were detected. As I mentioned, there was significant history of dementia in the maternal family, as we can see in the pedigree. Along with the proband, one other member has been confirmed to have the D209E mutation. On your right, you can see the protein, the presenilin-1 protein with the different hotspot mutations color-coded based upon different ages of onset. 209 is extremely close in proximity to 206, which is also another hotspot. At 209, we have 4 mutations, D209E, D209A, D209R, and D209V. All of these mutations have been reported in literature, but they have been characterized clinically, but none of them have any neuropathology reported with them. So we are the first study to describe the neuropathology affected with a G209E mutation. As you can see in the coronal sections, not only do we have neuronal loss, but we also have significant white matter involvement as outlined by white on these images at the level of the genu of the corpus callosum, and here at the level of the thalamus with the hippocampus right there. And as we progress to the antibodies against amyloid beta and then later to tau, we see a very interesting distribution of the severity of pathology as we go across the neocortex. Looking at a higher magnification, in G, we are seeing neuritic plaques and tangles in the amygdala, which might potentially explain the mood dysregulation and anger outbursts. At a higher magnification of the hippocampus, we see the severity of tau positivity affecting the different neurons, especially the hippocampus and around the CA1, CA2 regions. Looking at eye, we see tangles and neuritic plaques in the striate cortex, which confirms a Braak staging of 6. One interesting finding for this case was the presence of diffuse plaques and globose tangles in the oculomotor nucleus or the Edinger-Westphal nucleus, which is seen in panel J. Here you're seeing diffuse plaques. Not only were the neocortical areas involved, we also had the cerebellum have pathology as well. As you can see, diffuse plaques very abundant in the cerebellar molecular layer. You also have amyloid angiopathy visible here as well. In L, we are again in the cerebellum, looking at the microglia in the white matter, the molecular layer, and the granular layer. The final diagnosis for this case was a Braak stage of 6, SERA score C, and NIA Reagan high likelihood. Apart from A beta pathology and tau pathology, no other pathologies were noted. No antibodies against alpha-synuclein and PTDP43 did not show any positivity. To summarize, this is the first study describing the pathology associated with a mutation affecting codon 209 of presenilin-1. We have several interesting pathologies, especially the pallor of the white matter, of the myelin stain, the pallor of the myelin stain showing the white matter involvement, and of course the findings in the oculomotor nucleus and also in their involvement in the cerebellum with the spinocerebellar tract, eventually in the spinal cord as well.